For this particular video, we're going to concentrate a little bit on what's going to prepare you the best for biochemistry, just in terms of like a real brief introduction to a pretty important classification that we've been going through the last couple of days here called proteins. These things that are made up of amino acids. It turns out that pretty much all of life is pretty much dictated by proteins. So, along those lines, chemists, or I guess I should say more generally, scientists that study this kind of thing have divided different levels of protein structure that we have to understand. Most of what we're going to concentrate on is primary structure, but I just wanted to make sure that you kind of saw like the full context of the picture here. So, what we've been talking about in terms of amino acids thus far has everything to do with this idea that these things have a primary structure, right? And honestly, it's kind of weird to say primary structure. It's really the sequence of amino acids that you're talking about when you say that. So a great example is towards the end of the last presentation on Wednesday, we talked about a little peptide that was made up of things like glycine and phenylalanine. So it turns out if you were to say the primary structure of something, I'd know that you were talking about the exact sequence. That's where you would look at those little 3-letter codes or sometimes the 1-letter codes and determine what the actual amino acids are that make up that protein. Keep in mind that there are 20 naturally occurring amino acids, and there's a couple in different biological systems that are kind of derivations of those. But for the most part, all of the proteins in the universe are made up of those, or I shouldn't say the universe, the proteins that we know of on this planet are made up of those 20 amino acids. There's also this idea of secondary structure, and we'll just see some pictures of this. And it's due to hydrogen bonding, and specifically hydrogen bonding between what's called the backbone of a protein, right? That regular repeating nitrogen, carbon, carbon, nitrogen, carbon, carbon. The most common ones that you're going to be introduced to in either a biochemical or a biological context are known as the alpha helix and also the beta pleated sheet, though there are beta turns and random turns and a couple of other kind of oddball structures there. As you kind of continue to zoom out, right, the tertiary structure, and most proteins do have tertiary structure, though there are some structural proteins that technically don't have tertiary structures. That's really what's called the complete 3D conformation. I really think about this as like the boogery shape of a protein, right? When you look at, like, say, some sort of enzyme, it's always really important that you understand the way that all of those different folds and kinks in the chain kind of come together. And this is mostly due to interactions like intermolecular forces, or even sometimes disulfide bridges between side chains, right? The variable group, the R group of amino acids. And then sometimes you have to talk about quaternary structure, and this is something where 2 or more different proteins come together to form kind of a larger aggregate protein. A great example of this would be something like insulin or hemoglobin are really common examples. So let's kind of break this down just a little bit. First of all, we'll talk about secondary structure. We're going to kind of work backwards to kind of the more organic, specific primary structure stuff, just because it's not, in terms of like an overall context, it's not as important. So I'll kind of put it at the end of these slides here. So here's a great example of that alpha helix, right? A lot of people have already been introduced to helices because they know about the double helix in DNA. Even though we're not going to talk about nucleic acids very much, it's probably something you're familiar with. And so instead of being a double helix, this kind of alpha helix is actually it, by the way, has handedness, right? You can twist a helix in 2 different ways. And this thing is considered what's called a single helix, right? In other words, instead of having like DNA basically has 2 rungs of a ladder kind of twisted around each other with the base pairs as the rungs of the ladder. This thing just has like a single turn, and we'll see kind of what this thing looks like in a bigger protein in just a second. And keep in mind that these things are held together between hydrogen bonds of the backbone, right? Another really common structure is this thing called I learned it as a beta pleated sheet. They might have gotten rid of that nomenclature. But this is kind of, it reminds me a little bit of something like graphite, right? This idea of just having these different laminar layers, and this can occur locally in some sort of peptide as well. This isn't the best picture of a tertiary structure of a protein, but it is kind of giving you an idea of the important folds that happen in a protein, right? Like, honestly, I should include a different picture, and let's do that right now. So here are a couple pictures I stole from the internet. This one is just kind of a generalized artist rendition of the way that secondary structure can be part of an overall bigger tertiary structure here. So this is what the alpha helix might look like, right, when that backbone attains that conformation there. And obviously what happens is that you have all this other strand that doesn't really do much of anything, but then you'll have these various regions of alpha helix, and then of course you can also have these kind of anti-parallel pleated sheets here, and then occasionally these things are also labeled as beta turns. But keep in mind that this is always the part of, or not always, but if you're talking about a globular protein, which most enzymes are, you're talking about this big giant kind of assembly of amino acids where that has to be folded in some particular shape. In other words, structure really dictates function, right? If you don't have certain parts of these proteins folded correctly, this is where you get proteins that don't work, and that's usually the cause of a whole bunch of different diseases, whether they be genetic or even prion diseases and things like that that you'll hear about. This very next picture, I just kind of picked out a couple of other random pictures just to kind of show you the types of pictures you'll be looking at in biochemistry. This one just happens to be a type of cobra venom, or at least a peptide that's part of the cobra venom. And you can kind of see like the oftentimes where there's a beta pleated sheet, they might put some sort of kind of like an artist rendition, not necessarily artist rendition, but a computer generated rendition to kind of label certain beta types of assemblies. And then there's also this little alpha helix here, which I really can't see. They labeled a beta turn for you, which is another type of secondary structure. But obviously the tertiary structure is kind of the full assembly here. This other picture is just an important class of signal transduction proteins that are embedded into the cellular membranes of living systems. These things are called G proteins, which are often a part of various cellular responses and things like that. And so each one of these barrels is actually an alpha helix. Interestingly, like these things are made up of hydrophobic amino acids primarily because they have to fit in the bilayer here, but then you have these other hydrophilic things in both the extracellular and intracellular compartment. And these little subunits right here are usually involved in the signaling cascade. And so this is like the interior of the cell right here. So just kind of hopefully a little bit of context. I really like this summary slide just to kind of help you to understand what we're talking about with the different types of structures. Of course, the one thing that we didn't talk about is quaternary structure. And so I'm going to have a picture of insulin in just a second to kind of show you what's going on there. So here's a good example of something that's pretty common in a biochem course where you would talk about the quaternary structure of something. So insulin's actually a hormone. It's not actually an enzyme, but you can still see its quaternary structure here. It forms a couple of different polymers here, or I guess it's not called polymers. There's another name for like a complex, I guess, maybe for these enzymes. But anyway, you can see an insulin dimer, right? And you would say, well, this thing has a certain globular shape. You'd be talking about the tertiary structure. And then you could talk about the tertiary structure of this. And obviously there's some alpha helices in terms of secondary structure. But you could ask the question, what's the quaternary structure? And that's where you're talking about like the overall kind of globular shape of the dimer, or you can even have a hexamer. And so in a sense that this quaternary structure is something to know about, but certainly not all all proteins have this kind of thing. And the different kinds of interactions that hold these things together are the same kind of intramolecular attractions that you might imagine hold together a tertiary structure. Certainly, from an organic perspective, it's a good idea to talk a little bit about how we determine the primary structure of peptides. A lot of this is kind of historical, and things are done a little bit differently now. Your book does a really good job of going through all the different processes. I'm just going to kind of highlight a couple of the important things through the reading. First of all, it's a good idea to know a little bit about different kinds of chromatography, right? So this is probably from a high pressure liquid chromatograph. And so typically what you do is kind of a first step when you isolate a new protein is, and this is something that's even true today, is that you throw it into a big giant vat of acid, and then eventually all of those individual amino acids all break apart. And what you can do is you can put it through a special ion exchange column that where amino acids come out at really particular times, depending on what kind of protocol you're using. So, for instance, this is a pretty typical looking thing where you have a standard where you know exactly where different amino acids come out. You throw your protein into a big giant vat, and out pops all of the major amino acids that make that thing up. Of course, you don't know much about the amino acid sequence at this point. That's where different kinds of degradations are going to come into play, which we'll talk about now. So the first things you can do to get an idea of protein structure is this thing called partial hydrolysis. Your book has some really great pictures, or at least kind of a better look at the logic behind all these things. But again, it's really similar to the idea of a mass spec where you don't know the structure of something and you hit it with a big giant hammer and try to put together all the pieces of the jigsaw puzzle that are broken apart in certain ways. And so, for instance, if you had some sort of simple protein of 4 different amino acids, if you broke it up into, let's say 5 or 6 different fragments of 2 amino acids each, eventually you'd be able to look at those fragments and tell the exact sequence. There's also kind of fancier ways that, honestly, I could never remember in questions. For instance, that they would say, I degraded a protein with trypsin, and I could always remember that they cleaved at really particular amino acids, but I could never remember where. But it turns out there's 2 proteases that are really good at cleaving on the carboxy side of really particular amino acids. The trypsin cleaves obviously at the end of these basic type amino acids. So you would pretty much throw your protein that was unknown into a solution of trypsin or vice versa, and out would pop a bunch of different segments that always ended with lysine and arginine, and you'd be able to look at that thing, determine its structure. Kind of a similar thing, this other protease named chymotrypsin, except this thing cleaves at the end of the various aromatic amino acids. And honestly, a really common question, at least in my biochem class, which was taught by a small molecule biologist, is to remember exactly what these things cleaved at. So we get these really cool kind of Sudoku type problems where we have to determine protein structure, where I really would have no idea exactly where, like, what the information was telling me, because I could never remember which one of these cleaved at a particular site.